Hello, my name is Vedanti Gaikwad. My roll number is 6217. The enzyme which I am going to explain is maltase. So let us get brief introduction of the enzyme maltase. Maltase is a alpha glucosidase enzyme found in the small intestine's brush order. This enzyme catalyzes the hydrolysis of the disaccharide maltose into two simple glucose sugars. Its EC number is 3.2.1.20. Plants, bacteria, yeast, humans and other vertebrates have maltase. It is supposed to be made by cells in the mucous membrane that lines the inside of the intestine. Let us see history of maltase. The history of maltase discovery began when Napoleon Bonaparte declared a continental blockade in his Berlin Decree in 1806. This initiated the search for alternative sources of sugar. In 1833, French chemist Anselme Payen and Jean-François Persos discovered a malt extract that converted starch into glucose, which they called diastase at that time. In 1818, H.T. Brown discovered mucosal maltase activity and differentiated it from diastase, now called amylase. In the 1960s, advances in protein chemistry allowed Arne and Dacquelst and Giorgio Semenza to fracture and characterize small intestinal maltase activities. Both groups showed there were four major fractions of maltase activity that were intrinsic to different peptide structures: sucrase isomaltase and maltase glucoamylase. 50 years later, entering the genomic age, cloning and sequencing of mucosal starch hydrolysis confirmed Dacquelst and Semenza's findings. Structure of maltase. Maltase belongs to G813 glycoside hydrolysis family 13 that is responsible for breaking apart the alpha glucosidase linkages of complex carbohydrates into simple to use glucose molecules. The glucose molecules would be then used as a sort of food for cells to produce energy, adenosine triphosphate, during cellular respiration. Mechanism of maltase. The mechanism of all family GH13 enzymes is to break alpha glucosidase linkage by hydrolyzing it. Maltase focuses on breaking apart maltose, a disaccharide that is linked between two units of glucose at the alpha 1,4 bond. The rate of hydrolysis is controlled by the size of the substrate, carbohydrate size. Production of maltase. The enzyme starch is partially transformed into maltose during the digestion process by the salivary pancreatic enzymes known as amylase maltase. Maltase is secreted by the intestine and then converts maltose into glucose. The so produced glucose is either utilized by the body or can be stored in the liver as a glycogen, or called animal starch. Industrial production and applications of maltases. Alpha amylase has an important function in degradation of starches, so it is extremely common used in the baking industry. It is mostly used a means of flavor enhancing to improve bread quality. Without alpha amylase, yeast would not be able to ferment. Maltose glucoamylase is commonly used as a fermentation source as it is able to cut starch into maltose, which is then used for brewing beers and shake. Other than brewing, maltose glucoamylase has been studied by introducing specific inhibitors to stop the hydrolysis of the alpha glucosidase linkages. By inhibiting the cleave of the linkages, scientists are hoping to devise a drug that is more efficient and less toxic to treating diabetes. Thank you.